In this scientific paper titled, Globular Proteins and Where to Find Them Within a Polymer Brush, a case study, the authors explore the dynamics of protein adsorption on polymerized surfaces using a coarse-grained, CG model. The aim of the study is to understand the effects of various design parameters on protein adsorption efficiency. The introduction begins by highlighting the significance of studying proteins, as they are essential for life and have complex behaviors and interactions with their environment. Proteins fold into specific 3D structures, and their conformational dynamics are influenced by factors such as the presence of confining surfaces and surrounding molecules. The authors acknowledge that studying protein adsorption is challenging due to the vast number of possible configurations and interactions involved. Atomistic simulations, while accurate, are computationally demanding. Therefore, the authors propose using a coarse-grained model that simplifies the system while preserving the main microscopic characteristics. The chosen CG model is based on the hydrophobic polar, HP, model for proteins, where hydrophobic amino acids are attracted to each other and avoid water molecules. The authors explain that the HP model captures the tendency of hydrophobic residues to stay in the inner part of the protein, surrounded by hydrophilic, polar residues. The study focuses on the adsorption of globular proteins on a polymer brush, which is a surface made up of polymer chains grafted to a substrate. The authors explain that polymer brushes are mechanically stable and have wide applicability. However, there is limited research on the combined study of proteins and polymer brushes. The authors describe the setup of the simulation system, including the polymer brush with different grafting densities and the globular proteins with different hydrophobicity ratios. They also introduce variations in the system, such as the presence of attractive beads along the backbone of the polymer chains or the addition of ligands. Overall, the introduction provides an overview of the significance of studying protein adsorption, the challenges involved, and the chosen approach of using a coarse-grained model to explore the dynamics of protein adsorption on polymer brushes. The specific parameters and variations in the simulation system are also introduced. Rate and percentage of protein adsorption. This figure shows the rate and percentage of protein adsorption for different conditions. The x-axis represents time, and the y-axis represents the rate and percentage of protein adsorption. The different lines in the graph represent different conditions, such as grafting density and protein size. The results show that the rate and percentage of protein adsorption depend on these factors. Higher grafting density and smaller protein size lead to higher rates and percentages of adsorption. Effect of grafting density. This figure shows the effect of grafting density on the adsorption of proteins. The x-axis represents the different grafting densities, and the y-axis represents the height, radius of gyration, and end-to-end distance of the polymer chains. The results show that higher grafting densities lead to higher heights and larger dimensions of the polymer chains. This indicates that higher grafting densities result in more extended polymer coatings, which can affect the adsorption of proteins. Conformational properties of proteins. This figure shows the conformational properties of proteins in the absence of the coating. The x-axis represents the different protein types, and the y-axis represents the gyration radius and end-to-end distance of the proteins. The results show that the size of the proteins remains the same regardless of the hydrophobicity ratio or the presence of the coating. This suggests that the size of the proteins does not significantly affect their adsorption. Rate and percentage of protein adsorption as a function of hydrophobicity ratio. This figure shows the effect of the hydrophobicity ratio on the rate and percentage of protein adsorption. The x-axis represents the different hydrophobicity ratios, and the y-axis represents the rate and percentage of adsorption. The results show that higher hydrophobicity ratios result in lower rates and percentages of adsorption. This suggests that more hydrophobic proteins have a lower affinity for the polymeric surface. Effect of a bead position on protein adsorption. This figure shows the effect of the position of attractive A beads on protein adsorption. The x-axis represents the different A bead positions, and the y-axis represents the rate and percentage of adsorption. The results show that the position of the A beads does not significantly affect the rate and percentage of protein adsorption. Effect of A bead hydrophobicity and position on protein adsorption. This figure shows the combined effect of A bead hydrophobicity and position on protein adsorption. The x-axis represents the different A bead hydrophobicities and positions, and the y-axis represents the rate and percentage of adsorption. The results show that the hydrophobicity and position of the A beads have a minor effect on the rate and percentage of protein adsorption. Protein density profiles. This figure shows the protein density profiles for different conditions. The x-axis represents the distance from the surface, and the y-axis represents the protein density. The different lines in the graph represent different conditions, such as coating type and hydrophobicity ratio. The results show that the protein density depends on these factors. Higher hydrophobicity ratios lead to higher protein densities, especially close to the surface. Effect of grafting density on protein density profiles. This figure shows the effect of grafting density on protein density profiles. The x-axis represents the distance from the surface, and the y-axis represents the protein density. The different lines in the graph represent different grafting densities. The results show that higher grafting densities result in lower protein densities, especially close to the surface. Effect of a bead position on protein density profiles. This figure shows the effect of the position of attractive A beads on protein density profiles. The x-axis represents the distance from the surface, and the y-axis represents the protein density. The different lines in the graph represent different A bead positions. The results show that the position of the A beads does not significantly affect the protein density. Effect of hydrophobicity ratio on protein density profiles. This figure shows the effect of the hydrophobicity ratio on protein density profiles. The x-axis represents the distance from the surface, and the y-axis represents the protein density. The different lines in the graph represent different hydrophobicity ratios. The results show that higher hydrophobicity ratios result in higher protein densities, especially close to the surface. Potential of mean force, PMF. This figure shows the potential of mean force, PMF, for protein adsorption. The x-axis represents the distance from the surface, and the y-axis represents the PMF. The different lines in the graph represent different conditions, such as coating type and hydrophobicity ratio. The results show that the PMF depends on these factors. Higher hydrophobicity ratios result in higher PMF values, indicating a higher energy barrier for protein adsorption. Overall, the results of this study show that protein adsorption on a polymeric surface is influenced by various factors, such as grafting density, protein size, hydrophobicity ratio, and the presence of attractive A beads. Higher grafting densities and smaller protein sizes lead to higher rates and percentages of adsorption. Higher hydrophobicity ratios result in lower rates and percentages of adsorption. The position of A beads does not significantly affect protein adsorption. The density profiles of proteins depend on grafting density and hydrophobicity ratio, with higher densities observed for higher hydrophobicity ratios. The PMF for protein adsorption depends on coating type and hydrophobicity ratio, with higher PMF values observed for higher hydrophobicity ratios. Overall, these findings provide insights into the factors that affect protein adsorption on polymeric surfaces and can guide the design of surfaces with desired protein adsorption properties. Figure 12 in the paper shows the potential of mean force, PMF, for protein adsorption on different coatings. The PMF represents the free energy landscape that a protein experiences as it moves towards a surface. In this case, the PMF is calculated along the z-axis, which represents the distance from the surface. The PMF curves show how the energy barrier for protein adsorption varies with different system parameters. The significance of Figure 12 is that it provides insights into the factors that affect protein adsorption on the coatings studied in the paper. It shows that the grafting density of the coating and the hydrophobicity of the proteins play important roles in determining the rate and percentage of adsorption. The PMF curves reveal that higher grafting densities and more hydrophobic proteins result in higher energy barriers for adsorption, indicating slower and less favorable adsorption processes. Conversely, lower grafting densities and more hydrophilic proteins lead to lower energy barriers and faster adsorption. Additionally, the PMF curves demonstrate the impact of attractive A beads, located at the surface, and ligands, attached to the surface, on protein adsorption. These special sites can enhance or inhibit adsorption depending on their strength and density. For example, attractive A beads can facilitate adsorption by providing additional attractive interactions with the proteins, while highly attractive ligands can significantly increase the adsorption percentage. Overall, Figure 12 provides a comprehensive view of the factors influencing protein adsorption on the studied coatings and helps guide the design of coatings with desired adsorption properties. It highlights the importance of considering parameters such as grafting density, hydrophobicity, and the presence of special sites in controlling protein adsorption. The findings from this figure contribute to the understanding of protein surface interactions and have implications for various applications, such as biomaterials and drug delivery systems. Figure 13 in the paper shows the potential of mean force, PMF, for protein adsorption as a function of the distance from the polymer surface. The PMF curve represents the free energy landscape for the adsorption process, indicating the energetically favorable and unfavorable regions for protein adsorption. The PMF curves for different systems and conditions are compared, including different polymer grafting densities, hydrophobicity ratios, positions of attractive A beads, and the presence of ligands. The significance of Figure 13 is that it provides insight into the factors influencing protein adsorption on the polymer surface. The PMF curves reveal the energy barriers that proteins need to overcome to adsorb onto the surface. The shape of the PMF curve and the energy barrier height can be tuned by adjusting various parameters, such as the polymer grafting density and the hydrophobicity of the surface. The position and density of attractive A beads, as well as the presence of ligands, also affect the PMF curves and, consequently, the rate and percentage of protein adsorption. The PMF curves in Figure 13 demonstrate that higher grafting densities of the polymer coating result in higher energy barriers for protein adsorption. Increasing the hydrophobicity ratio of the surface leads to higher energy barriers as well. The position of attractive A beads and the presence of ligands can modify the shape of the PMF curve, influencing the rate and extent of protein adsorption. Overall, Figure 13 provides valuable information about the energetics of protein adsorption on the polymer surface and highlights the key parameters that affect the adsorption process. The results can guide the design of polymer coatings with desired protein adsorption properties. Figure 14 of the paper provides an illustration of the potential of mean force, PMF, for protein adsorption on different types of coatings. The PMF represents the free energy barrier that proteins must overcome in order to adsorb onto the polymer surface. The figure shows the PMF curves for different protein sizes, S25 and L25, and different types of coatings, mushroom, intermediate, and brush, for various attractive wall strengths. The significance of Figure 14 in the context of the paper is that it demonstrates how the grafting density of the polymer coating and the size of the proteins affect the rate and percentage of protein adsorption. The PMF curves reveal that the higher the grafting density of the coating, the smaller the rate of adsorption and the lower the percentage of adsorbed proteins. This is due to the increased excluded volume and free energy barrier for protein adsorption in densely packed coatings. On the other hand, the size of the proteins also plays a role, with larger proteins exhibiting slower adsorption rates and lower percentages of adsorption. Additionally, Figure 14 shows that the strength of the attractive interactions between the proteins and the wall surface affects the adsorption behavior. Higher attraction leads to an increased amount of adsorbed proteins, especially for lower grafting densities. The figure also indicates that the location of the attractive A beads, either in the backbone or as ligands, can influence the adsorption behavior, with different effects observed for different types of coatings. In summary, Figure 14 provides valuable insights into the factors that influence protein adsorption on polymeric surfaces, including the grafting density of the coating, the size of the proteins, the strength of the attractive interactions, and the location of the attractive A beads. These findings can be important for designing coatings with desired adsorption properties in various applications. Figure 15 in the scientific paper shows the effect of different parameters on the rate and percentage of protein adsorption. The figure includes two panels, one for S25 proteins and one for L25 proteins. Each panel shows the adsorption curves for three different polymer coatings: mushroom, intermediate, and brush. The x-axis represents time, while the y-axis represents the fraction of proteins adsorbed. The significance of Figure 15 is that it demonstrates how different factors influence protein adsorption on polymeric surfaces. In this study, the authors examined the effect of parameters such as grafting density, hydrophobicity ratio, and the position of attractive A beads on protein adsorption. The figure shows that these parameters can significantly affect the rate and percentage of protein adsorption. For example, higher grafting densities of the coating generally lead to lower adsorption percentages. Similarly, increasing the hydrophobicity ratio or the molecular weight of the proteins tends to decrease the rate of adsorption. The figure also demonstrates the importance of attractive A beads in promoting protein adsorption. It shows that systems with attractive beads, whether located in the middle of the backbone, at random positions, or as ligands, have higher adsorption percentages compared to systems without attractive beads. The figure further highlights the role of the strength of the attractive wall in protein adsorption, with higher attraction leading to higher adsorption percentages. Overall, Figure 15 provides valuable insights into the factors that influence protein adsorption on polymeric surfaces. It helps in understanding the complex interplay between various parameters and protein adsorption behavior, which can be useful for designing surface coatings with tailored adsorption properties. Figure 16 of the paper shows the potential of mean force, PMF, for protein adsorption on different types of polymer coatings. The PMF represents the free energy landscape experienced by a protein as it approaches and adsorbs onto the polymer surface. The x-axis of the figure represents the z-coordinate, which corresponds to the distance between the protein and the polymer surface. The y-axis represents the PMF, which indicates the stability or instability of the protein at different distances from the surface. The significance of Figure 16 is that it provides insights into the factors that affect protein adsorption, such as the grafting density of the polymer coating and the strength of the protein-polymer interactions. The results show that higher grafting densities of the coating lead to a higher energy barrier for protein adsorption, making it more difficult for proteins to adsorb onto the surface. Additionally, the strength of the protein-polymer interactions influences the adsorption behavior, with stronger interactions leading to a higher rate and percentage of adsorption. The PMF curves in Figure 16 also demonstrate the impact of protein size and hydrophobicity on adsorption. Larger proteins and more hydrophobic proteins tend to have higher energy barriers for adsorption, resulting in slower rates of adsorption. Overall, Figure 16 provides valuable insights into the factors governing protein adsorption on polymeric surfaces, which can be used to better design and control protein surface interactions in various applications, such as biomaterials and bioseparations. Figure 17 in the paper represents the asphericity values of proteins adsorbed on different coatings. Asphericity is a measure of the shape of the protein, with a value of 0 indicating a spherical shape and a value of 1 indicating a highly elongated shape. The figure shows that the proteins tend to remain close to their spherical shape, with slightly higher values for the adsorbed proteins on the highly attractive coatings. This suggests that the proteins are in a folded globular state, and their shape is minimally affected by the adsorption process. The significance of Figure 17 is that it provides insights into the conformational behavior of the proteins upon adsorption. The results indicate that the proteins maintain their native structure and do not significantly unfold or change shape during adsorption. This information is important for understanding the stability and functionality of proteins on polymeric surfaces, as well as for designing surfaces with desired protein adsorption properties. In addition, the figure also demonstrates that the hydrophobicity of the coating and the molecular weight of the protein can influence the shape of the adsorbed protein. More hydrophobic coatings and larger proteins tend to have slightly higher asphericity values, indicating a slightly less spherical shape. This observation suggests that the hydrophobic interactions between the coating and the protein can affect the protein conformation to some extent. Overall, Figure 17 provides valuable information about the shape and conformational behavior of proteins upon adsorption, which can aid in the design and optimization of polymeric surfaces for various applications, such as biomaterials and drug delivery systems. Figure 18 in the scientific paper provides an overview of the effect of different parameters on the rate and percentage of protein adsorption. The figure shows the fraction of proteins adsorbed, F_ads, versus time for different grafting densities of the polymer coating. The grafting density refers to the density of the polymer chains in the coating, which affects the freedom of movement for the proteins within the coating. The figure indicates that the grafting density of the coating is a significant parameter affecting protein adsorption. Higher grafting densities result in lower rates and percentages of adsorption, as the higher density creates a barrier that prevents proteins from easily getting adsorbed. Conversely, lower grafting densities allow for more protein adsorption. In addition, the figure also suggests that the size of the proteins and the hydrophobicity ratio of hydrophobic to polar beads in the protein, play a role in protein adsorption. Larger proteins and more hydrophobic proteins tend to have lower rates and percentages of adsorption. The significance of Figure 18 to the paper is that it provides insights into the factors that influence protein adsorption on the polymeric surface. The findings indicate that the grafting density, protein size, and hydrophobicity are important parameters to consider in designing coatings for protein adsorption. The results can be used to guide experiments and optimize the desired outcome of protein adsorption. Figure 19 of the paper shows the configuration of a polymer coating with a single protein tethered to the surface. The protein is represented as a yellow sphere, while the polymer chains are represented as gray lines. The figure illustrates the concept of primary adsorption, where the protein interacts with the bare, solid surface of the coating. The presence of the protein creates a barrier that must be overcome for other proteins to adsorb onto the coating. The significance of Figure 19 is to provide a visual representation of the system being studied and to demonstrate the mechanism of protein adsorption on the polymer surface. By understanding the interactions between the protein and the coating, researchers can gain insights into the factors that influence protein adsorption, such as the grafting density of the polymer chains and the hydrophobicity of the protein. This information can be used to design coatings with desired properties for applications in fields such as biomaterials and drug delivery. The paper also discusses other figures that are relevant to the study. Figure 6 shows the effect of the grafting density of the polymer coating on the rate and percentage of protein adsorption. Figure 7 demonstrates the influence of the hydrophobicity ratio on protein adsorption. Figure 8 examines the impact of the position of attractive A beads within the polymer chains on protein adsorption. Figure 10 analyzes the effect of the presence of ligands on protein adsorption. The paper also discusses protein density profiles, figures 11 to 15, the shape of the proteins, figure 17, and the potential of mean force, PMF, for protein adsorption, figures 20 to 22. These figures provide further insights into the behavior and characteristics of protein adsorption on the polymer coating. Figure 20 shows the potential of mean force, PMF, for protein adsorption on different types of coatings: mushroom, intermediate, and brush. The PMF is plotted as a function of the z-coordinate, which represents the distance from the coating surface. The different curves in the graph represent different attractive wall strengths. The significance of Figure 20 is that it provides insights into the energetics of protein adsorption on the different coating types. The PMF represents the free energy landscape that the protein experiences as it approaches the coating surface. A lower PMF value indicates a more favorable adsorption, while a higher PMF value indicates a less favorable adsorption. From Figure 20, it can be observed that the PMF curves for the mushroom coating are generally lower than those for the intermediate and brush coatings. This suggests that protein adsorption is more favorable on the mushroom coating. Additionally, it can be seen that increasing the strength of the attractive wall interaction leads to a decrease in the PMF, indicating a more favorable adsorption. These results suggest that the coating type and the strength of the attractive interaction play important roles in determining the rate and percentage of protein adsorption. The mushroom coating and stronger attractive interactions promote higher rates and percentages of protein adsorption. On the other hand, the intermediate and brush coatings, as well as weaker attractive interactions, result in lower rates and percentages of adsorption. Figure 21 in the scientific paper shows the potential of mean force, PMF, as a function of the reaction coordinate, z-coordinate, for the adsorption of a single protein onto a polymeric surface. The PMF represents the free energy landscape of the adsorption process and provides information about the energetics and barriers involved. The figure includes PMF curves for different systems, including different coating types, protein sizes, hydrophobicity ratios, and the presence of attractive A beads or ligands. The colors in the figure represent different attractive wall strengths. The significance of Figure 21 is that it demonstrates how different factors, such as the grafting density of the coating, hydrophobicity of the proteins, and the presence of attractive sites, affect the adsorption process. The PMF curves reveal the energy barriers that proteins need to overcome to adsorb onto the surface, and the differences between the curves for different systems provide insights into the factors that influence protein adsorption. The results show that the grafting density of the coating has a significant impact on the rate and percentage of adsorption. Higher grafting densities lead to lower rates of adsorption due to the higher excluded volume and energy penalty for proteins to access the coating. The size and hydrophobicity of the proteins also affect the adsorption process, with larger and more hydrophobic proteins experiencing higher energy barriers for adsorption. Furthermore, the presence of attractive A beads or ligands can enhance or inhibit protein adsorption, depending on their position and strength of attraction. The PMF curves demonstrate how the presence of these attractive sites affects the energy landscape and the adsorption process. Overall, Figure 21 provides valuable information about the factors influencing protein adsorption on a polymeric surface, allowing for a better understanding and potential control of this process in various applications. Figure 22 of the paper shows the effect of different attractive wall strengths on protein adsorption in different polymer coatings. The figure specifically focuses on the adsorption percentage of S25 proteins for different coating types: mushroom, intermediate, and brush, and different attractive wall strengths. The results show that the adsorption percentage is influenced by both the coating type and the strength of the attractive wall. For the mushroom coating, there is a considerably higher amount of proteins going in and out of the coating compared to the other coatings. As the strength of the attractive wall increases, there is a tendency for an increase in the adsorption percentage, especially for the lower grafting densities. This trend is similar to what has been observed in previous studies. The figure also indicates that the adsorption rate and percentage are affected by the grafting density of the chains in the coating. The density of the coating sets a barrier that proteins need to overcome to get adsorbed. Higher grafting densities tend to slow down the adsorption process, while lower grafting densities facilitate faster adsorption. Overall, Figure 22 demonstrates the significance of different factors, such as coating type, attractive wall strength, and grafting density, in determining the rate and percentage of protein adsorption on polymeric surfaces. These findings can be used to design and optimize coating materials for specific applications, such as biomaterials and drug delivery systems. The discussion and conclusion of the scientific paper summarize the findings and significance of the work conducted. The authors classified protein adsorption on polymeric surfaces into three categories: primary adsorption, secondary adsorption, and ternary adsorption. Primary adsorption refers to the attraction of proteins to a bare solid surface. Secondary adsorption occurs at the outer surface of the coating to avoid free energy penalties caused by protein insertion. Ternary adsorption takes place within the coating itself due to monomer protein attraction. The authors conducted simulations to study the rate and percentage of protein adsorption, as well as protein density profiles, for different scenarios. One major finding is that the grafting density of the coating significantly affects protein adsorption. Higher grafting densities restrict the movement of proteins within the coating and create a higher free energy barrier for adsorption. The size of the proteins also influences the rate and percentage of adsorption, with larger proteins experiencing slower rates of adsorption. The hydrophobicity ratio of the system also affects adsorption, with more hydrophobic systems leading to lower rates of adsorption. The authors also studied the shape of the proteins and found that they tend to remain spherical, with only slight deviations from their near-spherical shape. The presence of attractive beads along the polymer chains influences the shape of the proteins, with proteins tending to stay closer to these attractive sites. Overall, this work provides insights into the various factors that influence protein adsorption on polymeric surfaces. The findings have implications for designing coatings with desired adsorption characteristics and can contribute to the development of anti-fouling strategies and biomaterials. If you are an author or copyright holder for the work presented in this video and you have any issue with the existence or contents of this video, please let me know.